Hola. Hello. Well, thanks for coming. It's great to have Frédérik Saltré giving a talk in our Friday seminar series. Fred already gave a talk yesterday at the annual Congress of the museum. So for those who already were there, you will hear my introduction twice. So Fred is an ecologist and a biogeographer. Modeling biodiversity and environmental climate change is transversal to everything that he does. He currently holds a joint position with the University of Technology, Sydney, where he is a senior lecturer. And he's also a scientist of the Climate Solutions Group of the Australian Museum. He is also the chief investigator of the modeling node of the Center of Excellence for Indigenous and Environmental Histories and Futures. He's published more than 50 papers, more than 5,000 citations. We met in 2013. My PhD supervisor at the University of Adelaide became his postdoc boss at Flinders University. And we've been collaborating ever since, mainly on quaternary megafauna extinctions. Fred gave a talk yesterday on the out of Africa dispersal of anatomically modern humans. Today he will be touching on a completely different topic, I guess, the conundrum, if I can call it that way, of native versus non-native species in conservation science. Ready? Over to you.
Well, good afternoon, everyone. Thank you for having me again. So before you get too sick of seeing me, I'm going to try to talk about something a little bit different compared to yesterday. Leaving a little bit aside human expansion and going more into trade space and divergence in invasive and non-invasive species. So this work that I'm started to do came from an interesting conversation that I had with my daughter a few years ago, actually. That was funny. I remember that we were just having dinner and she asked me about extinction and species going extinct. I was completely shocked. That was the first time in forever that she even paid attention to what I was doing for a living. So we start talking about environment, biology, and extinction. And those are the species that pretty much went extinct during my lifetime before she was born, so before 2007. And so that's pretty much this species. She didn't know what it was, other than in a picture, because they don't exist in wildlife anymore. And for her, that kind of makes sense, because I'm old, so that's normal that those things happen. But then we move a little bit more, and those are the species that were extinct during her lifetime. And so those are the species that she knows about, but when she's going to have children, she's going to be exactly in my shoes, not being able to show them other than on pictures. And so we had this conversation about species extinction and what's going on at the moment. And whether or not we agree that we're in a 6 mass extinction context, well, the summary is pretty grim when we look at the state of the planet, with 50% of vegetation of the biomass lost, more than 70% of land surface altered, usually by us, 40% plants are endangered, more than 700 vertebrate and 600 plant species have been extinct just for the last 500 years. 2/3 of the ocean are compromised by human activities, and 1 million of species are threatened with extinction in the near future, and just 1 million of species that we know of. So to do something about it, there are several options. One, that's a tool that has been used, and you know that, I think, all too well, has been rewilding. So I'm not going to explain you in too many details what the rewilding is. You're all too familiar with it. But pretty much that's the idea of getting away a little bit of the species in itself, but more focusing on the role that the species and the function that the species is going to have on its ecosystem and trying to restore those things. So there are many different forms of rewilding. So from the Pleistocene rewilding, where we try to reproduce or come back to a former baseline by introduce other species to get a past baseline for the ecosystem, or passive rewilding, where you just leave nature going through its own way without us having too much intervention. Or trophic rewilding, that's the textbook case of apex predator trying to stabilize the ecosystem, or translocation-based rewilding, that you put back a species that used to be there and wasn't anymore. Those, no matter what we're looking at, it's all about always reintroducing species that are not there anymore in a new context. And there are more or less success with those strategies. But when we're trying to apply that to Australia, the place where I'm from, there's a little bit of a different consequences on that. The main idea is most of the rewilding options means that we want to introduce species that are not in Australia. And so the non-native species that we're talking about come from Northern Hemisphere in general. Unfortunately, we have another name for that. We call that invasive species, because most of the species that have been introduced from Europe in Australia became invasive. That's an example here for the rabbit, where as soon as we put the rabbit in the ecosystem, it spread everywhere. And so that's the habitat suitability for the European rabbit in Australia. And the problem is that it outcompetes all of the native species. So it has huge consequences. Over the last 60 years, the Australian government spent more than $390 billion in trying to manage the impact of invasive species in Australia. So it's not small feat to have. So in that context, the rewilding part of it, introducing which species should we introduce, mostly from Northern Hemisphere, in Australia, has to be thought very carefully. Still, disregarding this impact, we start seeing a little bit of a push in Australia from a school of thought saying that we should actually embrace those invasive species, cats, foxes, deer, and pigs, because they indeed can help facilitate other species and sometimes replacing lost functional services. So in this talk here, even if intuitively we can think, maybe not, the idea is it's never been formally tested. And so in this talk, I'm going to start presenting some of the results that we've been working on, testing this idea of can introduced species actually be a solution or be used as a surrogate for invasive, for native species. So because this talk is about 30, 40 minutes long, just in case, the short answer is no, we can't. So you know exactly where I'm going there. I'm just now explain how we get there. So when we want to start talking about ecosystem resilience and functional diversity or functional services, it starts with traits. So any sort of feature that a species has to cope with its environment. So there are plenty of different traits, database with traits. So we're talking about weight, diet, locomotion, length. So all of those for different type of taxa, whether it's mammals, whether reptile, and so on and so forth. I'm going to focus here for Australia on mammals only, but the framework that I'm presenting can be applied to many other taxa. So let's have a look at the mammals in Australia first, and then I expand that a little bit later. So we take all of the traits, 18 different types of traits, from the time of activity, diet, breath, limb, litter size, locomotion, and so on. We all have them. For each species, we have trait values. And we're going to group mammals in Australia in 4 different groups. We're going to have the non-native predators, native predators, non-native prey, and native prey. So those groups have been made, nativeness and non-nativeness, relative to European colonization in 1788. So that's why the dingo is in the native species, even if it's not fundamentally a native species in Australia. So we have those 4 groups. And the idea is to analyze what are the difference in trait, in function, between those 4 groups. If non-native species could replace native predator, for example, you take non-native predator here and native predator, they should theoretically have the same type of functional diversity or trait space, being you can replace one with the other. To test that, let's imagine here that that's the trait space. So each dot's going to be one species, one individual, all right? I'm looking at just 3 axes here, the habitat breadth, the body mass, and the time of activity. We use as a human to think in 3 dimensions, 4 sometimes if we are lucky. Beyond that is a bit too much. Then we try to localize those groups. So here's going to be, for example, your non-native species here for the predator. This one's going to be all of the species that's part of the prey, native prey, and this one is native predator, for example, and so on. In real life, it's just for, as an example, is never that clean, the differences. There are a lot of overlaps, but just for the sake of explaining the logic, that's how we go with. Once we have that, you imagine here that you have 3 axes. You're going to have that for all of the different traits. So we need to find a way to summarize that in some sort of metrics that we can actually understand and interpret. It turns out that geometric morphometry does that very well. They call that disparity analysis. So usually geometric morphometry, if you're familiar with that, they have skulls and they measure a bunch of different measurements and try to summarize the information. And so they summarize the information in 3 main ways. The first thing is going to be the size of each blob. So the blob that I call the blue cluster, for example, that you saw before, or the red cluster. We're going to compare those things. Do they have the same size? Do I have the red blob bigger than the green blob, for example? In ecological terms, that's what we're going to talk functional diversity. We talk about functional diversity. The bigger, the more function you have, all right? The more variance in the traits you can have. The second that you can have is for the same size, you can have difference in density. So for the same size that you have, do you have something extremely packed, or do you have a lot of holes in there? So that's the ecological equivalent of functional redundancy, all right? And then you have the position. Position you can have in different ways. You can have overlap between 2 blobs, or the relationship between and the distance from one blob to the center of the big cloud. So that's what I'm going to use here in this talk. So relative to the center of the big cloud, the closer you are, the more trait you share with the rest of the group. So you're much more of a generalist in terms of function. The more on the margin you are, the more of an outlier or kind of specialist or vulnerable you are, all right, in this logic. Here is the result of the comparison of the position relative to the center for all of those 4 groups. And as you can see here, so the higher up you are in this axis, the further away you are from the center. So the more of a specialist you are. 2 things that you can look at here. When you compare the cats and the dingo, so non-invasive predator and non-invasive predator, they have nothing in common. So you can't really functionally substitute one by the others. When you look at the prey here, exactly the same. So so the first thing is you have a higher variance and a higher specialization for the invasive species in Australia compared to the native. Now that we see that we have difference in functional in function and functional diversity, which trait is responsible for that? Is it one trait? Is it several traits? So to try to rank the traits important, do we have similar traits that are responsible for non-native or for native? So to rank that, we're going to do what we call a sensitivity analysis. So it's a pretty simple way. You have your density value for the benchmark, so for one group, let's say non-native species, native species, for example. This group is going to have a value for position with confidence interval, for the size, and for the density. And then what you're going to do, you're going to remove one trait, and that's going to shift those 3 metrics. Remove another one, that's going to shift the metrics again, and again, and again, and again. Every time you're going to have a departure from the benchmark, and you're going to measure the distance with this departure for each trait that you remove. The bigger the distance, the more of an impact this trait had. All right? So it allows you to rank which trait matters the most. I'm going to simplify this graph. I just put that here because I love spider plot, and I've been dying to use the opportunity to have a spider plot on the presentation. So here it is. You have here all of the, in blue, that's the representation of the ranking for each of these traits. The closer you are of the margin, the more important the trait is. So in blue for the native species, in red for the non-native species. So if you simplify, those traits are the one that matters in the comparison. Let's simplify even more. Those 3 traits are the ones that are important for the native species, and those 4 traits, 4 or 5 traits are the ones that are important for the native species. So we have winning age, body length, and habitat breadth for the native, diet breadth, locomotion, digit numbers, trophic level, and brain mass for the native. Again, completely different traits. So we want to try to replace predator in Australia with species that are actually nothing in common. What we can do, what we did here, you can do it for the prey as well. So long story short, if you look, there is a little bit of a different story when you look at the prey, because the pattern looks the same. The pattern looks the same, but the sensitivity is different. If you look at the yellow bit, which is the non-native one, it's much more sensitive to the green one because you're further away from the center. So you have a sensitivity that even if the trait seems to be, you seem to see with the herbivore, for example, or prey species, not much of a difference. In terms of magnitude of change, there is a difference. And I'll dig into this a little bit later. So, the resolution is not so great. Anyway, so if you compare those traits, so body length, brain mass, and winning age, and you do that, that's horrible, okay. And you have the same for prey species. What this figure tells you beautifully is pretty much that in between those traits, you have non-native species that tend to be bigger than the native one. They're brainier, so their brain is bigger as well. It's not just, not necessarily because of allometry, they tend to be bigger. They win earlier, and they reproduce slower. So it's completely different biology as well. Now, when you look at, in terms of trophic level, digit, and locomotion, that's the proportion of those traits for each group. The only thing that I'm not going to go through this graph too much. The only thing that I want you to see is how uniform everything is with the non-native one. We just have one type of trait. So in that case, you're going to have omnivore, for example, for the trophic level. For the digit, you're going to have claws only. And for the locomotion, you're going to have terrestrial only. What you have here for the native species, it's much more different. See, you have more carnivore, omnivore. You have much more differentiation. So the idea here is your invasive species, non-native species, are much more specialized. So again, you try to replace a species that is not that specialized, much more of a generalist, by something that's hyper-specialized and technically much more vulnerable or much more unstable if you remove it. When you're looking at the prey species, the main difference, if there is over here. That's the amount of insectivore that you have as a native species in Australia that you don't have with the invasive species in Australia. And insectivore in Australia tend to be keystone species for the ecosystem, which is something that you cannot reproduce with the non-native one. What we did to rank the species, the traits, we can do exactly the same ranking the species. Which species drive more the signal within your communities? So you have a rank here for the invasive species, for example, where you have the fox that represent the most of the variation in the functional diversity, then cats and dogs and so on and so forth. For native species, you have mostly those two species that matters. But the interesting part is not so much the ranking here, is the value down there, is the order of magnitude that you have in difference of sensitivity. So you have here with the non-native species, a system that is highly sensitive to any sort of changes. If you remove the fox, for example, you change completely the ecosystem very quick. If you remove one of those species, a native species in Australia, the native one, the system doesn't change that fast. It's much more resilient. So if I summarize just what I said for the last few slides, we're trying to, are non-native species a functional surrogate for extinct sometimes, or for native species? So short answer is no, because you try to replace something that's pretty stable and quite resilient by something that's hyper-specialized and can generate unstable ecosystem for once. It's mostly because Australian species are the result of 1,000 years of evolution, because it's really isolated, and there is a long story of evolution that you don't have with the non-native species. And actually, we also have some keystone species that are not being replaced by those new arrival. So that shows you the need, with this kind of framework, the need for having a baseline somehow. All right? So the main question is there any sort of, we start having a hint that whether or not we have some traits or combination of traits that's going to make species more resilient or less resilient, and which are they in Australia? The thing is, everything that I showed so far was at Australian scale. But spatially speaking, it must be much more heterogeneous. So you have regional variation. So can we apply this kind of framework at a regional scale? So instead of looking at invasive species, not invasive species, then this time you can group your cluster here based on location. So you grid the space, and instead of having invasive, non-invasive, prey, and so on and so forth, you're going to have the red location, the blue location, the green location. Exactly the same principle, and you can have exactly the same metrics and compare them. And you start having then some maps. You can map the size of each cluster by location. You can map the density as well, and you can also map the position. So the idea being, when you have a low size, so you're more in the light colors here, that means that you have more identical traits and a lower adaptive response to threat, for example. Same with the density. So if you want to interpret that in terms of chance of redundancy, for example, the lower the density, the lower the chance of redundancy. With the position, when you increase the position, so if you're in the dark blue here, you're going to have more outlier, so more chance to be at the margin, much more vulnerable in your ecosystem, for example. So you can spatially explicitly have those kind of baseline. And again, I spent a lot of time trying to do this animation sensitivity analysis, so I try to have some sort of return of investment here, and I show it again and again and again. But each grid cell, you can have this sensitivity analysis as well, and rank which trait matters the most per location. And so that gives you a map like this, where you have which traits dominate the whole system. So in that case here for Australia, you're going to have here the trophic level. That's really important. Here you're going to have the time of activity, for example. You might have the hibernation of torpor here, for example, and so on and so forth. If I summarize this, we pretty much have all Australia being shaped by the time of activity, torpor, and the trophic level, all right, in terms of traits. Then you can compare that to any sort of environmental factors. Environmental factor in terms of precipitation, in terms of accessibility to the terrain, human impact, agriculture or whatever, connectivity. Do I have my community, are they endemic, just in one spot and nowhere else? Or do I have a big range of community, or what is my nearest neighbor, for example? Do I have any sort of connectivity? Fire, a big thing in general, mostly in Australia, and temperature. So we're trying to see which of those factors best explain that pattern. And so I gloss over all of the statistical stuff that goes underneath, because we can talk about that later, but it's not very talk-friendly. So believe me, there is a lot of math behind that, usually model comparison for each grid cell and stuff. What it shows is mostly this pattern, and the pattern of the traits that we're seeing is mostly driven by maximum temperature, human impact, and also precipitation level. All right. So temperature, when we look at the maximum temperature and now the trait space shift in terms of more specialized species or more generalist, we have this kind of threshold here between 20 and 25 degrees in maximum temperature, where you have some sort of a threshold where if you're below that, specialists tend to try to conserve a little bit of energy. It's getting a little bit too cold, a little bit too constraining for Australia, at least, We're talking about maximum temperature. Maximum temperature in Australia usually is much higher than that. 25 is pretty chill. And so to limit energetic demand, some species tend to specialize a little bit more into the torpor side of it. So that's the relationship between what's happening in terms of climate, maximum temperature, the trait, torpor, and the location that I showed you before. In terms of precipitation, that's a different story. We have some sort of a U-shaped pattern between generalist and specialist, where you have many more generalists in your community here, and at the sweet spot around 100,000 millimeters of total precipitation a year. And below that and above that, you go more towards specialist-driven or rich community. So whether it's because the resources here, you get a little bit too dry, so the resources are a bit more scarce, so you need to adapt a bit more, or you have some sort of niche partitioning here when you have too much precipitation. And then finally, the landscape type that you have, which is completely dependent on human activity, for example, in Australia. But here is much more about aridity, and because the landscape type, those two here, that have the most of an impact are the ones that are completely dry and barren in the middle of the desert and in the outback. So in that case, what the species needs to do is to regulate their time of activity to be able to cope with this kind of constraint that they have. I did that for Australia. Why stop there? Let's get greedy a little bit and do that for the rest of the world and see if we have something similar from Australia compared to the rest of the world. So you can also have, doing exactly the same approach that I did before, map of functional diversity based on this framework for the entire world. Same with functional redundancy, and same with the position. So that shows some pattern that I'm sure you're familiar with. And with the same thing, you can have then the main dominant traits for all of those grid cells. Exactly the same thing. I didn't put back the sensitivity analysis approach where the things that are moving, I thought that twice was enough. But that's exactly the same thing. So you can see here that we have about 49% of the whole area that's mostly driven by time of activity. So whether it's diurnal, nocturnal, and so on. You have about 18% that's still based in terms of trophic level, mostly around here, and so on. And still a little bit of hibernation and torpor playing mostly here, and here. Same thing here. We can also look at what the climates look like and doing exactly the same thing. And we have some pattern, some biogeographic pattern coming here with precipitation. So precipitation being a huge factor mostly around here. Having the connectivity, at least, that we tested, that are quite surprisingly, in the whole northern hemisphere, the connectivity between your communities are quite important. And you have also here, minimum temperature. The thing interesting here is when you look at the pattern of North America here and the pattern of Australia is not as uniform as everywhere else. And so the question is, what's the difference? Why do we have something that's relatively uniform here, and here, and something completely different here and here? Well, there is an aspect that I was interested in considering was the time aspect. Do we have any sort of legacy in terms of climate change over time that could impact the species and the community as we see them today? So in other words, do we have some sort of legacy, climate change legacy, that would make your community more prone to respond in a way or another? So what I try then is to add into the analysis temporal changes in temperature and precipitation, in that case over the last 7,000 years, to look at, pretty much how much of change did we have over time for each grid cell in temperature here and precipitation. Okay, those two maps represent temperature here changes. So the more in the red you are, the more changes you had in terms of temperature over the last 7,000 years. All right. The more you're in the yellow, the fewer changes you had. Same with the vegetation, with the precipitation. Sorry. The more you're in the dark blue, the more changes and fluctuation in precipitation you had over time. The more you're in the light yellow, the fewer of the changes. All right. Those are simulated value, obviously, over time. And so this time, I had exactly the same thing as before, doing a model comparison analysis, testing the impact of precipitation, ruggedness, impact of human connectivity, fire count, and temperature. I just added this time the temporal effect of past temperature and past precipitation variability. And what you see here is, while you still have a huge effect of species connectivity in the northern hemisphere here, you still have precipitation in Africa, and you still have minimum and maximum temperature in South America. Those areas where we had those differences have shifted this time. And this time we have an impact, much more of a legacy of what happened in terms of temperature. So we can, I don't know, that can be called like some sort of climate burden that you can have spread over time. So if I summarize, I just wanted to present in this topic today a new framework that we use to define species traits. Usually in species trait, I mean, if you're familiar, we always use 1 or 2 dimensions of the entire trait space. The approach that I showed you used the entire variation of the trait space, using disparity analysis to summarize everything that's happening. And based on that, you can see that first, using non-native species for replacing native species can work. I'm not saying it's not working in certain contexts. I'm saying that in Australia, it's so different that for now, it doesn't seem to be working. The differences are way too big. And that allows you to create different type of baseline to make those sort of assessments in an objective way for everywhere around the world. So with that, I'm happy to take any question, and thank you for your attention.
Thank you, Fred. So we have some time for questions. Diego.
Yeah, so I have thanks, Fred. I have 2 methodological questions. Hola? Okay.
Yeah.
So I had 2 methodological questions and one more ecological if there's time.
Yeah.
So I really got this a clarification when you talked about shifts from a center.
Yeah.
What's the ecological interpretation of that center?
Well, the center is pretty much you gather all of the traits that you have in an entire with all of the species that you can have. All right. So you're looking at the amount of I would say overlap between traits. So when you're at the center, that means that you're actually sharing a bunch of similarities with the other species around you.
Okay.
All right. So it's more like I said, you try to. I don't like too much. I simplified a lot for saying that it was more generalist of specialist. Yeah. That's not exactly true. Okay. That's more how much of the variance and of the traits you share with other species relative to how unique you are when you're more on the margin.
Okay, I understand. And then the other methodological question was about, I mean, since you use the full trait space, I don't know how many dimensions that is, but did you encounter issues like as the dimensionality increases of the trait space, you start having a more sparse space, right?
Yes.
And could this sparsity represent an issue when trying to calculate metrics? I don't know. It's something it's not super clear in my mind, but I kind of have this intuition that if this the dimension starts growing a lot, yeah, this sparsity can introduce some I don't know if artifacts or
you can inflate a bit your metrics.
Okay.
To a point. The main reason I'm doing this, and I didn't want to bore you to death with the detail of the approach, but it's the problem that I see actually with the main approach that we're using now, which is based on 3-dimension or 4-dimension, you know, is first usually because everything is nonlinear, you represent about maximum 30% of the variance, which means that you neglect 70% of it. In the context of climate change, when you're going to have something now that probably doesn't really matter because it's dimension number 25, let's say, now doesn't mean that it's not going to matter in the future. And this dimension 25, if you don't account for it, you're going to miss it. So that was the whole idea. So it's some sort of a trade-off between, yes, you might inflate a little bit the metrics to a point, but you also have more information that you would have otherwise. So it's just, yeah.
Okay, fair enough. And then regarding the non-native species and the introduction in
Yes.
Australia, I mean, I get the message, but then it got me wonder, okay, what if we analyze from a pool of species that are from whatever hemisphere, the ones that do have traits that match? Then would you be okay with that? I mean, it's more of a question.
Well, there is more to it, all right? And I think that's and there's actually a conversation that we had yesterday, should be, at the minimum, something that should be done and tested, which is not. Which is just at the moment, we're just saying that, oh yeah, they look the same, they have the same body mass, they have the same trophic level, so that should be probably a good invasive species. My point being, we have so many endemic species in Australia, so many vulnerable species, the consequences are so big to include that it should be thought carefully. Right. And to a point, it's a lot of effort and commitment into doing this, to taking a species. I mean, translocating a species is not a walk in a park in general. It's costly, first, logistically. It's extremely stressful for species. So I think that's money better spent trying to protect and care for the species that we already have than trying to include other things. But that's just an opinion. My point being, we need to have much better tools to test what would be the long-term consequences. And I haven't even talked about interaction here. Yeah. All right? That this framework is, at this stage, is not necessarily a good one to test the interaction. We need to go a little bit different level. But if already at that stage you see differences, when you start going to put interaction into the mix, I would just slow down things a bit. That's the take-home message, I would say.
Okay, thanks.
Well, I have a question myself. So trait space interacts with ecosystem structure and function at a given point in time. Okay. Yes. So from a trait perspective, how much time back you go to consider species that went extinct native?
Yeah, the temporal. You're thinking in terms of traits, kind of evolution or stuff like that, or things like that, right? That's the only thing that we can do now at the moment is taking into account trait variation. So for one trait, we don't just have one value. We have a value with confidence interval that we account for in the analysis. So by resampling some stats trick, but this aspect that you're talking about is not included yet. And at that, at the moment you can't have both. You have some disparity analysis, for example, that shows the change of traits over time, and you talk in terms of evolution, but you don't have that across space. Or you can have it across space, but you cannot integrate that over time. I mean, at least it hasn't been done this way. So it's a really good question. My short answer would be, I don't know. But you're right that there's going to be a point where you need to include this kind of evolution of traits because it's going to change things. Thanks.
Thanks, Alba. Thanks for the nice talk. I have one main question which relates with the last one, and then a couple of technical questions. But first, let's go to that. Because perhaps it's because I'm in origin a dung beetle ecologist, and I know the history in Australia, and actually that dung beetles were imported to handle things that the local scarab fauna wasn't able to do, okay, or to decompose the cow dung. So you said that you haven't analyzed that, but to what extent the networks of interactions are decoupled between the invaders and the,
There are quite a bit, actually. The network are extremely different. And even in terms of in terms of relative importance, for example, when you rank those species, like I showed before, for example, if you look at the dingo, you don't have it at the top at all. So meaning that if you remove the dingo, yes, he has an impact, but you don't have this hypersensitivity to the system. And that's the main thing that I try to express is that you just, by putting those species here, that we're not talking about the same trophic level. I mean, a cat's not going to have the same diet as a dingo, for example, or the wolf, exactly the same. They kill pretty much everything, you know. So you don't include in the ecosystem this kind of high instability that you're going to create. Yeah. And it's not just a matter of time. It's not if you leave 5,000 years to the cat to adapt that I would change anything. It wouldn't. Because they're just really different because the position in the network is different.
Okay, so it could be the case that you remove some invaders and you collapse a module of invaders, but not the nature.
Yes, yes,
okay.
That's exactly what's going on.
Yeah. Okay, excellent. And then going a bit to the methods,
yeah.
Okay, because it's presenting simplifications is nice, yeah. But I wonder whether, I mean, in those, I personally don't like maps with only one factor in each place because of what, as you know, because it may be the case that the second best is always operating everywhere.
Totally
so
agree.
Are the big wheels that you were presenting representing the number of cells where that factor was the main factor, or the overall importance?
No, that's the overall importance, spatially, explicitly. Then we have a ranking as well per grid cell of which factor, which is pretty interesting to see if usually you have the same ranking. Yeah. So when you have precipitation first, you tend to have the second one is still the second one. Yeah. So there are a few occasions where you have a tie, or not almost, but it's pretty close. But usually you have a pretty big demarcation. But something that I haven't had the time to plot is would be the difference in ranking, for example, when you have. So for the same, if for two different grid cells you have the same first one, do you still have the same hierarchy after that? And
but in general, the spatial distribution of the importance mimics the overall importance of the factors. Yeah. Okay, and then the last one is about why are not because you're focusing on single traits, and I've worked a lot with trait diversity, of course, but usually the approach is just you focus on the hypervolumes. Yeah. And then you check what happens with the traits. Yeah. And you seem to have done it in a pretty similar way, but then you are putting a lot of emphasis on the specific traits rather than on the overlap between the trait spaces of the individuals. And there are some recent frameworks, I mean, or Llorente Culebra's papers on island mammals, or Tom Matthews' papers on global birds. Have you tried to check whether this overlap, how much the invaders overlap with the...
we're doing that with birds at the moment. So I haven't done it for mammals yet, but we're starting to try to look at, in terms of overlap, so we would have the position one, but instead of looking at the relative distance toward the center, looking at the relative distance between each other. Yeah. So that would be the.
But you're comparing distance between centroids are not whole overlap.
No.
Both.
I agree. No. But we're starting to do that. Yeah. For birds, we haven't had the result yet.
Yeah.
But yes.
Okay. Thanks. Yeah.
Any other question? Cool. Okay, so we finish here.
Thank you very much.